Hello class, welcome to our last week of lab. We are going to essentially do some tightening up and just sort of give last thoughts today. Um, I'm also today going to talk to you about the lab report, and you'll notice here that I've put that in quotes because it's not a traditional lab report. It's more of an extended set of discussion questions, but it is more of a final set of questions that really test the concepts and the ideas that we've been talking about all term in the lab. So what I'll do today is I'll first, just for old time's sake, review the experimental design, make sure that you're comfortable with what we've done and why, and remind you about how we've measured amyloid beta via the ELISA and what role the cell lysate played in our whole experiment. Then I'm going to show you the Excel spreadsheet, which is where the raw data are typically deposited when we do this experiment in the teaching lab. And I'll just give you a sense of how it's structured and the logic behind its organization. I'll then talk to you about how I've gone about normalizing the data, running the t-test, and then producing the graph. We'll also review the graph so we're comfortable with what it's presenting and how. I'll then actually go through the lab quote-unquote report, and remember, it's essentially an extended set of discussion questions, but I'll go through them with you, give you some insight into my thinking on those questions, and I won't answer them for you, but I'll certainly help point you in the right direction, so it'll be relatively easy to answer those. And then I'll have some final thoughts related to the lab. Okay, let's get started. So one more time, and you ought to be able to draw this in your sleep as I can. Let's talk about exactly what we were up to this term. So remember that we were interested in the proteolysis of APP. APP is the amyloid precursor protein. It contains this amyloid beta sequence, which is known to contribute to the plaque pathology in Alzheimer's disease. And it really correlates nicely with the areas of the brain where there is massive cell loss. And so people have been very interested in studying this fragment and its origin. So we know that the way we get amyloid beta out of APP is through cutting. Proteases, enzymes that cut proteins, will act as molecular scissors to cut this substrate ribbon. The beta secretase is going to cut first, and remember what it produces is the N-terminal fragment plus the C-terminal fragment, which contains the intact amyloid beta. Gamma secretase, the next protease, will cut here, and that will liberate the amyloid beta fragment. And remember that most of this is happening in the trans-Golgi network, and A beta gets packaged into a secretory vesicle, mobilizes to the cell surface, and then is deposited into the extracellular space. And that's really the origin of the plaque, because as we age, the ability to turn over amyloid beta declines. It's very sticky to begin with, and then some other processes may impact its stickiness. And then it, of course, forms that large insoluble aggregate. Now, we were interested in lab in this alternative pathway involving alpha secretase. And what's interesting about TACE, or alpha secretase, is it cuts within the amyloid beta sequence. So it still recognizes full-length APP, it just cuts in a different location than the beta secretase does. And the products of alpha cleavage include these two pieces, where the first part of amyloid beta goes along with the original N-terminus, and the other part of it goes along with the original C-terminus. And of course, that precludes the formation of amyloid beta because we've essentially cut it in half. Gamma still cuts here, but now we produce a different product called P3. This is secreted, but it's non-pathogenic, it's not sticky, it gets turned over very easily, even in aged individuals, and so it doesn't really present a problem. Just as a side note, from a therapeutic perspective, what most of the drug companies are interested in is inhibiting beta secretase activity. And the logic there is if you inhibit that enzyme, you sort of artificially force more of the APP to be proteolyzed by the alpha secretase that's available to cut it. Now, we took a sort of different approach. One of the things that we were interested in is seeing if we could shift the competitive balance between these enzymes by driving the pathway in this direction. We did that by activating a signaling pathway which upregulated alpha secretase activity. Note that the levels of alpha secretase were not impacted, so the same amount of protein was present. We just caused it to be more active. We did that by exploiting a known signal transduction pathway involving protein kinase C. And what we did was we stimulated it with PMA, and then that activated PKC. That in turn phosphorylated TACE and fully activated it. Remember that TACE is activated in kind of a two-step mechanistic process. First step is proteolysis, removing the inhibitory prodomain. That happens during maturation of the enzyme when it's moving through the secretory pathway and gets into the Golgi. And that proteolysis of the prodomain removes the inhibitory influence of that prodomain on the active site. Now the active site is exposed and able to engage its substrates, including APP. The second half of the mechanism involves flipping the molecular switch up. We talked about it being like the rheostat or the dimmer switch for a light bulb. You turn the switch on, and that's the binary action of the proteolysis, by way of analogy. And then you move that switch, you slide it to a higher position to make the light glow brighter. That's analogous to the phosphorylation by PKC. So that two-step process fully activates it. And what we did see, was that when we did this in CHO695 cells, we detected lower amounts of amyloid beta in the conditioned media after PMA treatment. And that supports our hypothesis that we were able to shift this proteolytic balance in favor of the alpha side. And of course, because the substrate is limited, there's only so much of it left to be proteolyzed by beta and gamma, and that's why we get a lowered amount of amyloid beta. And that, in a nutshell, was our experimental design for the class. Treat with PMA, collect conditioned media, perform the A-beta ELISA. Okay, remember why we performed the ELISA. The ELISA is a sensitive technique to measure a specific protein of interest. And for us, it was an amyloid beta ELISA. And so, we had a well that was coated with the amyloid beta capture antibody. We added our conditioned media sample, and then the media contained amyloid beta, which stuck to that antibody. All the other proteins in that conditioned media don't stick. They just sort of remain in solution in the ELISA well. We came in with a detection antibody. That's why we call it a sandwich ELISA. We're sandwiching the peptide of interest, A-beta. And then we did some other antibody with HRP, added a substrate, and then we got a colored product that we could measure at absorbance of 450. So that's the basic idea of the ELISA. We're using the conditioned media because the main species of amyloid beta is secreted. And so, of course, we could have measured it inside the cells, but there really wouldn't be much point to it. That's a very minor species of amyloid beta. The major species that's produced is going to be secreted. And besides, that's the more relevant population to the disease. Now, we did collect cell lysates. Remember, we washed, after taking off the conditioned media from our plates, we washed, we then added the lysis buffer, scraped, and then did the sonication and centrifugation. And then we measured total protein with the BCA assay. And remember, the BCA assay, bisynkininic assay, is a colorimetric assay that relates to the reduction of copper in the presence of a basic medium. And then that cuprocyan reacts with BCA to give us a colored product. And there's a linear relationship between the amount of the colored product and the amount of the protein present. And we did that because we wanted to know roughly what is cell number in the well. We could count those cells, but it would take us a lot longer than just simply measuring total protein. Total protein's a good proxy or approximation of cell number because cells will have roughly the same amount of protein inside of them. So therefore, the amount of total protein in a well is directly related to the amount of cells in that well. And the reason we wanted to collect the total protein levels was we wanted to basically divide for a well our amyloid beta data by our total protein. And this is a way of normalizing to cell number, and it ensures that any reduction in amyloid beta that we see is not due to simply fewer cells in the well that the PMA treatment occurred in. So it's a way of just making sure, even though we wouldn't expect there to be pipetting errors when we're setting up these plates, we just want to make sure that we do our due diligence. And whenever we do science, we always need to carefully control and carefully account for every potential variable, even though they may be unlikely. So that's what we did. We did the ELISA for amyloid beta using the conditioned media. We did the BCA assay of the cell lysate to basically create this ratio. And that's what I will talk about when I show you the data in just a sec. All right, guys, we're looking here at a screenshot of my Excel spreadsheet, and I just, because of file sizes and other complications, did not want to import the actual Excel file into this software I'm using to do these lectures. But the screenshot will be fine, I think. So let's blow it up a little bit and see what we're looking at here. This is from last fall. So last fall, this is fall of 2019, I ran this lab with 12 groups, so 24 students, 12 pairs. And you can see that I just started off the spreadsheet by entering the group numbers here. We just assigned each group a group number. Okay. And then what you can see, I'm just going to get it, so you can see all of the lysate data here. There we go. So this side of the spreadsheet, this is columns B through S, is the total protein calculated via the BCA assay. So remember, when this is run in the lab, we start the ELISA, and then while the ELISA is incubating, we perform the BCA assay. And then the students get a printout that they bring to a TA, and the TA enters it into this master spreadsheet. So I've sort of broken it up where over here, these are the minus PMA treatment data, and over here the plus PMA treatment data. Remember the experimental design. We had a 6-well plate. We cut the plate in half. This is the minus, this is the plus. We labeled them 1, 2, and 3 for each. So here is sample 1. And remember when we do, so that represents the lysate from this well. When we do the BCA assay, we do it in duplicate. So here are the duplicate values for group 1. And here's the average. And those units are in mgs per mil. Okay, that's the milligrams per mil in the cell lysate. And usually anywhere between 0.5 and 1 is typical for when students do this in the teaching lab. And this one's at 0.623. So that's pretty typical. Then here are the data for well 2. So the cell lysate from this, again, in duplicate in the average shown in yellow. Same thing for sample 3 for the minus PMA. And then over here, the plus PMA, sample 1, sample 2, sample 3, again, same logic, duplicates, average, duplicates, average. This one, for whatever reason, didn't work. Duplicates, average. So then what we do is we calculate the average of all the student groups minus PMA lysate samples. And you see I just kind of obscured it, but it's 0.669. Okay. And then we do the same thing for basically taking the average of all of the plus PMA for the entire class, 0.625. And that does not seem different at all, and again, it's not. Look at the t-test on this, 0.623. So it would be weird if there was a difference here, because we would not expect to have plated these wells any differently. We'd expect the cell number to be the same, and because we have essentially the same mean, I mean, it's minor, very minor difference, doesn't even come close to approaching statistical significance, we know that there really shouldn't be any influence of cell number. Now we'll still normalize every group's individual A-beta data to their own lysate data, but I always like to run the t-test on the whole data set for the class just to show you how there's essentially no effect overall. And that's across 12 groups, so that's quite a powerful test. So that's what we've done there. We've entered all those data, and that's what we're going to end up normalizing to. Okay? So now what I'm going to do is I'm going to move the spreadsheet over so we can see the amyloid beta data. So now what we're going to be looking at is the calculated concentration for A beta. And that's here in columns T through Y. And again, I've sort of split it where the minus group samples 1, 2, and 3 are over here, and then the plus PMA groups samples 1, 2, and 3 are here. Remember, these are the conditioned media ELISA data. The ELISA is read in units of picograms per mil, so you can get a sense of just how sensitive the ELISA is. We're down to the picogram level of detection. And so for group 1, sample 1, we had 210 picograms per mil of amyloid beta, 177 for their second and 138 for their third. And there's some variation there, but that's not atypical. And it's better than some of the other groups that you'll see if you kind of look down that column. You can see it's a little bit all over the place. But this is a very good result. I remember them being technically very sound. Here's another group that was actually very good. So 236, 163, 156. These are very typical numbers for the A beta calculated concentration. Okay, so then let's look and see what happens when we add PMA. So here you can see we've reduced the M1 of amyloid beta. This group had, this range of data for their 3 samples, 98, 84, 89. Let's look at this group. Well, they've dropped these out, but 56, yeah, I won't really count that. It's not too impressive, but here's another very good one. 206, 167, 165, and then dropping it down pretty dramatically with PMA. And this is pretty typical, guys. This is what we typically see. Here's a group that had really consistent levels here, 100, 101, 103, and you can see, look how beautiful their technique was. Look how perfect these are, 71, 76, 69. And whenever I see that that's a group that had very good hands, very good technique. So we can take all of these data and produce averages and take a look at it. Let's do that. This is not normalized data, okay? This is just taking the raw data. But I always like to do that because it gives us a point of reference. So let's take a look at the raw data. For the entire class, the average amount of secreted A beta for minus PMA was 122. For the entire class, the plus PMA was 76. So that's a very typical reduction. And you can see, the standard deviation and the standard error, so we get a sense of the variation in the data, and the p-value with a t-test. And so, that's obviously remarkably significant. So that's the unnormalized data. Now, that's informative, and certainly given that we didn't see any real difference across the entire class in the lysate data, I think that that's believable. But I always like to go the extra step and normalize each group's samples. So on a sample by sample basis, do that normalization. So what we do here, now this is the average of all those normalized values, but I want to give you a sense of how this is calculated. So I take group 1, I take their minus PMA sample 1 value, 210, and then divide it by their value for their minus 1 lysate. Okay? And then we do that for the second sample and the third, and then we do that for the plus PMA data as well. And what that gives us are these normalized concentrations. Okay? So all of these data points are accounting for cell number. They are normalized to total protein. And of course, we can calculate the average of that for both the minus and the plus. And you can see that, we get about 2 is our unit here, 2.03 or 2.04, and over here 1.35. So clearly that reduction is maintained, and that's not surprising. So running the t-test on these data, we are at 0.0489. So we're at p of less than 0.05. And you can see how it drops the p-value pretty dramatically, right? So here we're at, 10 to the minus 6th. And here, obviously we're at 0.05. but significance nonetheless. And again, it's always better to account for variation. Okay, so the last thing is to prepare this to put the data, the normalized data, in a relativistic relationship so that we can more easily look on a graph at the magnitude of the reduction. So what I do is I divide each of these values by the average for the minus PMA group, and that's what's shown over here. And do that all the way down, get all these values, and when I average them, it gives me 100. What I'm essentially doing here is setting the control minus PMA treatment to 1 or 100. So then I can express the plus PMA as a fold increase or decrease of that. Do the same thing over here. Take these values, also divide them by this average. And again, that's expressing the plus PMA essentially as a percentage of the minus PMA. And so we can see that we end up with 100 for the minus PMA, we're at 65 for the plus PMA. And that is the magnitude that we will end up graphing, and I'll show you the graph in just a moment. So last step is to calculate standard deviation and then standard error, so we have a sense of the variation in the data. And just a quick note, the t-test is the appropriate test because we're comparing two means. If we were doing more than two means, we would use analysis of variance or ANOVA. The type of t-test that we're doing is we're doing an unpaired t-test and we're doing a two-tailed t-test. And the reason for that is we don't want to assume any direction of the change. We want to always have the most conservative test that we can run. And remember, anything less than 0.05 is our threshold for significance. Okay, that's about it for the data presentation. So, I don't want you to get too deep in the weeds on this, but I do want you to get a sense of how we might handle these kinds of data. The most important thing to take from this is that we're taking our amyloid beta data and normalizing it to the BCA data. And that's really the most important point. And that's going to, again, make sure that we're doing our due diligence and taking account for any unanticipated variable that may be playing a role here. And then what you get after the graph, of course, is a visual representation of the magnitude of the PMA effect. Let's take a look at that now. So here it is, and remember what I did in the previous slide. I showed you how we set the minus PMA to 100, and then we just looked at the plus PMA as a percent decrease of that. And that's a very typical effect for an entire class full of students. The error bars that we're presenting here are the standard error of the mean, and that's a good way to represent the data because it helps us to understand the variation within the data. A good rule of thumb is when error bars don't overlap, you are probably into statistical significant territory. Not always. It's certainly not always the case. I want to give you the impression that that's always the case, but as I say, a good rule of thumb. And we are here at 1 star. By convention, 1 star is p of less than 0.05. 2 asterisks would be less than 0.01. And 3 would be less than 0.001. And we don't really go beyond 3. We just kind of leave it there, typically. And so you can see what I've done here. I have written a figure title. Your figure title is really the bottom line result. You want to declare the result. And there would be a lot of different ways to phrase this result. And on the lab report, I will ask you to come up with your own title and figure legend, and I want you to put that in your own words. These are my words. Upregulated alpha secretase activity reduces A beta secretion. What you try to do is you try to bottom line it. You want to give the precise conclusion that we can draw from the graph. Clearly there's a reduction. It's statistically significant. That's the reduces A beta secretion part. And we know that the PMA treatment upregulated alpha secretase activity. So that's why that's in there as well. And then the figure legend. And the figure legend, I just want to speak a little bit about that. You should think of the figure legend as kind of a microcosm of the experiment that produced the figure. So you want to have a little bit of model system, a little bit of methods, a little bit of precise methodology in that figure legend. You also want to introduce any symbols that are not indicated in the key. For example, the asterisks that I do here. and you want to always report how the data are presented. So what I mean by a microcosm of the study, you have to think temporally or sort of chronologically at how the study was done. We started with the plate of CHO cells. We split them into two groups, minus and plus. So we treated them with PMA, and we always give the concentration, 1 micromolar, for 2 hours. That tells the person reading this how the experiment was done without them having to refer to the methods, if this was a paper, for example. We always want a figure to stand alone, and we want the figure legend to give us enough information to interpret the data shown, have a sense of how the experiment was run. So then I say after the 2 hours, we harvested the conditioned media and performed the ELISA. Those levels were then standardized to total protein as measured by the BCA assay, and then the stats were run with the two-tailed t-test to show significance. Data are reported as mean plus or minus standard error of the mean. So what I'd like you to do is to try to put that in your own words, and of course you're going to be guided by the template that I've provided here. That's not unusual or problematic, but you need to try to encapsulate those elements of the study with your own words. And the best way to do that is to spend time, and by the way, the best way to do any kind of answering of questions when you have a template or lecture slides or a book available for referring to. The best way to answer things so that you avoid inelegant paraphrasing is to absorb the information. So make sure that you're fully understanding the information that's presented, and then give yourself a break of, a day, come back, and then attempt to write out the information without looking at the template. And that last part is really key. If you do try to write the answer while you are looking at the template, you're going to probably paraphrase in a way that may step over the line. So let's try not to do that. Let's actually try to absorb the information, give it a rest so it percolates around your brain, and then you come back and try to write it without referring back to the source material. That's a good way to do our academic work, and it helps to ensure that we don't have instances where we unintentionally cross over some boundaries. Okay, I am essentially done with the data analysis part. Now we'll move into the instructions for the lab report. Okay, so this is the lab report. This has been uploaded to Moodle. Or if it hasn't, I forget whether I've done it or not, to be honest, but it will be there certainly by Friday, by tomorrow. And I just want to kind of go through this with you and give you insight into my thinking, okay? And I'm not going to answer the questions for you, but I'm going to help you think about where to look, how to think about them, essentially. There's a couple reasons I've chosen to do it like this. Initially, I had thought about having you do a more traditional lab report, but I think given all of our unique circumstances this term, and by the way, let's hope that this never gets repeated in our lifetimes, my God. I really felt that giving you something a little more structured and a little less open-ended would be help to alleviate some anxiety that you may be feeling, and I certainly am feeling. So let's try to be human with each other and make things a little simpler. Yeah, that's the idea. So that's why I put this in quotes. This is not truly a lab report, but it's a lab quote-unquote report. Okay. And what I've told you here, just so there's no confusion, the report that you're completing takes the place of the lab discussion questions I had planned to post for weeks 8 and 9. So you don't have to worry about that, and there were never going to be lab discussion questions for week 10. So what you do here will complete your work in the lab. In other words, this is the last lab assignment. Don't look for more, don't expect more, no matter what the syllabus may say, whatever. This is it. I'm telling you that now. Hooray. So you have until Friday of week 10, 5 o'clock local time, and that's because that's what I promised you in the syllabus for this lab report. Frankly, I considered moving that deadline up, but I don't want to create confusion or anxiety, so I'm leaving it in place. But here's my advice. Please don't wait until week 10 to do this. Right now, you've just heard this lecture. It's all fresh in your mind. Spend half an hour, whatever, doing it now. Okay? Do not wait. Just knock it out, get it done, upload it to Moodle, and don't think about it again. You've got other stuff going on in your lives right now. Don't agonize over this, okay? So, my advice, do it early. Do it as soon as you finish listening to me talk right now, and just knock it out. There is absolutely no legitimate reason to wait until week 10. Then if you wait till then, you're going to be 2 weeks removed from, really engaging with the lab material. And just don't do that. Don't put yourself in that position. Do it now. Work on your own, not in groups, okay? This is the way we've done all of our assignments. And, feel free to look at the recorded lectures again, read the manual, etc., whatever. Consult with your TAs, your professors. Write succinct responses. I'm not expecting huge essays for any of this, okay? By the way, when I write the key and give it to the TAs, it's like 2 or 3 sentences for each question, right? So I'm not expecting you to write a novel here. All right, let's go through. Question 1 says to outline the 3-part organizational scheme for the lab. This is basically the overarching organization that I talked about in some of the early lectures. And I want you to, think about that and why we had organized it that way and try to indicate what the purpose of each part was. So here I'm not talking about the hypothesis testing. I mean, it's part of the organization, obviously. What I'm talking about is how the entire quarter was organized. Okay, and you'll remember there was some skill set development, bioinformatics, and then some hypothesis testing. That's what I'm talking about here. So think about what the purpose of each of those parts were and why we organized it that way. That'll help you answer that question. Part 2, question 2, briefly outline the hypothesis we tested in lab this term. By now, this is something that should be second nature to you, so I don't think I need to go beyond that. You just need to outline that hypothesis in experimental design that we tested in lab this term. I'd like for you in part 3, or question 3 rather, to talk about the choice of the CHO 695 cell line. Why was that an appropriate choice? And it has as much to do with the fact that the CHO cells are easy to deal with as it does to the 695 designation. So make sure you understand what that's getting at and why that's an important, appropriate choice of model system. 4 is just basically, hey, what's conditioned media? You ought to have a sense of that through our lectures. 5, what's a lysate? Again, you ought to have a sense of what that is. 6 kind of relates to the lysate. It's asking us to say, hey, what's the difference between BSA and BCA, other than an S in the middle position of the acronym, right? Don't answer it that way, but try to give us a sense, and don't just define the acronym. Actually go into it and say, hey, what's the purpose of the BSA in that assay, and what's the BCA doing in that assay? 7 is looking at this relationship between alpha secretase and TACE, and we talked a lot about that in the bioinformatics lecture, in the bioinformatics lab. 8, 2-part activation mechanism and the role of PMA in PKC. It's just basically helping you, guiding you to some things I talked about earlier in this lecture. How do we get TACE activated? The bioinformatics lecture will also, and lab will also contain my thinking and instruction on that. 9 and 10 relate to APP proteolysis and the data that we collected. 9 says, name another APP cleavage product we could have analyzed to test our hypothesis. So you need to look at the cleavage schematic and think about what other cleavage products may be appropriate to test our hypothesis that we did, or that we tested this term. And then I ask you to give a prediction for that product following PMA treatment. What would happen to it? Would it go up? Would it go down? Would it not change? What would happen? 10 is basically the writing component of this lab report that's not answering a question. And it says basically give me a figure title and a figure legend for the figure that appears on the next page. And of course it's the one that I just went over. And it says be sure to put the title and legend in your words, and I've given you some instruction on that as well. I'll move over to the next page just to drive home the point, and then that's really all I have to say about the lab report, and then I'll give you some final thoughts. Okay, as promised, here's that figure. So you're going to write a title and a figure legend for this figure, and you're not going to copy the one that I gave you. You're going to put it in your own words. All right, folks, so some final thoughts. What I've tried to do for you, with you, this term in lab is give you a sense of what I would call project-based science. This is akin to what you would find if you went into someone's laboratory and were working with them on a research question. And the project-based approach is really different from what I call the modular approach that we may find in other courses. And there's nothing wrong with the modular approach, but at some point in our training, we have to transition more into holistic project-based approaches. So the other thing I've really tried to do for you here is to take a single project that we explore over time. And we start off by practicing, and then we inform our hypothesis. We do some hypothesis testing, and then we do some analysis. And this is exactly what we do in the lab, in a research lab all the time. And when you're working in a lab, you're not just running one experiment, one technique, you're really stringing things together. So that's what I've really had you, at least virtually, do this term. So the last thought that I'll leave you with is that, we obviously are operating, all of us in society, not just us in this course, under a very different set of circumstances, and while not ideal by any stretch of imagination, I hope at least somewhat effective. Honestly, it's disappointing to me that we were not able to do this in person. And while I feel that the experience you've gotten, I certainly hope, I feel that it's at least a semblance of what I would normally do. It simply isn't possible to recreate this experience virtually. It's frustrating to me, and I can only imagine it must be very frustrating to you as well. So I want to offer that anytime in the future, at some point, we will all be together again on campus. If at any time you'd like to come in and chat with me about this project or the work, I would really enjoy that. I would love to be able to sit with you and talk about the project that we've tried to do here. Okay, so that's it. We're done with lab. Congratulations and good luck. And again, please do stop by anytime to chat with me next time you're on campus, and I am. And I hope that's in fall. Okay, my friends, take care, and I will hope to see you soon. Goodbye.